For those of you who don't know us, so I run a small company in Seattle called Phase Genomics. And what I'll do today is I'll tell you guys about the technology. Some of you have worked with us before, but for those of you who haven't, I'll introduce our tech. I'll show some published, unpublished data of some of our projects. And then at the end, I have sort of a call out for a new project we're doing for which we're looking for collaborators. So the problem that we're trying to resolve is one that's very known to everyone in the audience, has been addressed by lots of speakers. The fundamental problem that when you're dealing with any kind of metagenomic sequencing, you extract DNA, at which point it all gets mixed and you lose a lot of valuable information. And of course, one of the casualties of shotgun metagenomics is that if you have mobile elements like phages, like plasmids, other things, you sort of lose the contiguity, their connection to the genome where they came from, right? And there's all sorts of other problems, right? You're trying to recreate genomes for multi-chromosomal organisms is extremely difficult, strain deconvolution, all these sorts of things. And so, so we've harnessed a sequencing trick that was originally designed for something else. This is a technology called proximity guided. So it's called proximity ligation. Some of you have heard of it, called it Hi-C. You guys saw Yuna's awesome talk. That was an example of it. So it was originally developed to look at three-dimensional folding of the genome. And the way it works is you basically take cells, you fix them with formaldehyde, you treat them with formaldehyde, and what that does is it goes inside the cells and it creates physical connections, basically covalent junctions between molecules that are physically next to each other, right? And what that does is it traps in physical space DNA sequences that are next to each other, which you can then basically recover and create this proximity ligation. It's a little bit like if you've ever tried cloning a plasmid, right? And you want the plasmid to ligate stuff, but it wants to close on itself. That's basically what proximity ligation is. DNA sequences that are close together are preferentially going to ligate. And then what that does is it creates these chimeric junctions. And so then you sequence this red-blue chimeric junction, and you know that whatever the red sequence is on the left and whatever the blue sequence is on the right, they were physically close to each other in three-dimensional space, right? And this is what's used to do the 3D modeling that Hi-C is known for. But remember, the cross-linking happened at the very beginning of the experiment before you broke the cells, right? So these junctions happened inside the cells. And so whenever you have two sequences that are physically interacting by this assay, they must have started out inside the same cell at the beginning of the experiment, right? So if we have a metagenome, right, you will get junctions both intra and interchromosomally, right? You'll get physical connections between the same chromosome and between different chromosomes. And that'll tell you sort of which sequences started out inside the same cells. And so you can use that to deconvolve metagenomes. And importantly, the junctions they're all not just inside the same chromosome, you also get ones between them. So if you have a plasmid or a phage, it'll cross-link to the genome of the host, and you can actually connect mobile elements to their host at the same time. So we've shown a number of ways. So one of the things that this does, this trick, it allows you to basically supercharge your mag binning, right? So when you're deconvolving metagenomes, right, you're trying to use tetranucleotide frequency, co-abundance, these sorts of things. Now you have another piece of information, which is which contigs have been touching each other, right? Which ones came out of the same cell. And so that sort of supercharges your ability to create sort of high contiguity, high accuracy mags. We have a platform for doing this called Proximeta. It's really good for getting into that dark matter of the microbiome because it doesn't require sort of databases. It just says contig A, contig B were touching each other, put them into the same mag. But the other thing is more relevant, I think, for this conference is we've shown, as well as other labs, that you can connect mobile elements to their hosts at the same time as you're doing this mag binning, right? So the same data set can be used to basically, on the left here, we're talking about antibiotic resistance and plasmid genes. And then a group in Paris showed back in 2017, you can do this with viruses and phages as well. So, right, so here's your mags that you've assembled. And then here are antibiotic resistance genes, right? So what's cool is it doesn't have to be a one-to-one relationship. So if you have a stripe like this, that means whatever this gene is, it's touching all these different hosts, right? And if you have a stripe going across, that would tell you that, you know, I don't know, let's say this one here, where you can see blocks going this way, that would tell you that this particular host contains all these different AMR genes. So You can detect multi-drug resistance. You can detect plasmid host range and things like that, sort of broad host range type mobile elements. And the same thing with phages, right? You can place them to their host without any culturing, without having to do, without having to do any isolation. And so then we pushed it a little bit one step further. The idea is that if you can say, okay, this is a phage contig, which mag does it belong in? Who is it touching? But if you have two contigs in the metagenome that both seem to be viral, and if they're touching each other and they're also touching the same bacterial host, that argues that they're probably part of the same phage. Actually, this can be used, these two pieces of information can be used to not just connect phages to hosts, but actually reconstruct phage genomes, make little mags for phages de novo, right? So now what we're doing is we're creating mags for the microbes, we're creating mags for phages, and we're connecting them together at the same time. We've done a bunch of validation, I'll show you in a second, but basically it looks something like this. So, this line here, that means there's a number of contigs that are touching each other, and they're all coming out of the same microbial host. And so you say, okay, well, this is our viral mag, okay? And then you can actually connect them to their bacterial mags in sort of large numbers. So what you're seeing here is on the x-axis, these are your phages, and on the vertical, on the y-axis, these are your microbial mags. So you can see how many of them we can get out of just a single, this is a fecal sample, I think. So every one of these bleeps in the radar, that's a phage and who it's infecting. And you can also see there's quite a few of them where there is a sort of a stripe like this. That means that there's a number of different phages infecting the same microbe, right? And then if you have a vertical stripe, that means you have phage that can infect multiple different bacterial microbial hosts. And this isn't limited to bacteria. This method will put together archaea, it'll put together eukaryotes, etc. So we now have a way of reconstructing phages and connect them to their hosts without culturing directly out of metagenomes. We've done a good amount of validation. This is basically comparing these little viral mags to long read assemblies where the whole phage is a single contig. So like sort of how accurate are we doing? And it's in this particular example, 19 out of 25 were perfectly supported by nanopore data. Another several were like when you have two phages inside the same host, then sometimes it confuses the binner. But in general, it's all been it's all sort of much more accurate than I expected at first. Of course, the neat thing is this works well with Illumina data. So like long reads are great, but this way you can get a lot more mags for your money. And there was a paper that came out last year from Tan Zhang's lab in Hong Kong. And what they've done was they compared, they were doing CRISPR-based annotation. It's a CRISPR annotation-based host affiliation of phages. And they needed an orthogonal data type, which they compared to. And so what you can see in these little pink and green boxes that essentially in cases where we had sort of overlapping sequences, it was over 90% sort of in agreement with the Proximeta data. And so it agrees, so the Proximeta data agrees with the CRISPR data, with the CRISPR annotations. But then if you look here, so the main difference is, right, so they were able to find like 20 things with the CRISPR-based methods and like 2,000 with the Hi-C. So it's just, it agrees when you have sort of the same ones, but just the amount you get with just the shotgun sequencing with Hi-C method, you just get so many more of them. And then it's extremely powerful when combined with long reads. So this is a paper we published earlier this year. This is a sample. This is a single sample. It was, I believe it was a sheep poop. It was sequenced pretty heavily with PacBio. And then also we did our Proximeta thing. This one sample had something like 1,200 mags. Over 400 of them were more than 90% complete. 200 of them were resolved into lineages. So basically like platinum, like strain level phasing. Importantly, there were over 400 phages, 300 plasmids, and tons of AMR host connections at the same time. And this is all, so this is what you get if you're willing to kind of pour money into a PacBio machine. And so what it does is right now we can do cool experiments, right? So here's a time course. This is a pretty simple experiment. It's just it's a recovery. It's a time course, a recovery from antibiotics. So time 0 is basically the last day of clindamycin treatment, and then you're kind of recovering from it. And as you can see, you're getting population, like more microbes are coming into the population, more viruses are appearing, right, over time. At the same time, plasmids and AMR genes are going away, which is what you would expect, right, with recovery from antibiotics. But remember, because for every one of these samples, we have this proximity data, now our time course can look like this, right? Well, we're now looking at phage host interactions over time and how are they changing. So not only appearing and disappearing, we can see them switching hosts, we can see them spreading to multiple hosts, we can see new things coming in, et cetera. And at the same time as we're doing this with phages, we can also do the same thing with plasmids. Just for brevity, I'm not showing those slides, and more. So here's sort of a more practical example. So a lot of the work we've been doing lately is in fecal transplants, right? And what's cool here is we are simultaneously, we can watch MAGs, we can watch antibiotic resistance, we can watch phages. So on the left, this is antibiotic resistance genes and who their hosts are, okay? And so you can see there's sort of two major AMR hosts in this sample, E. coli, Klebsiella. If they're yellow, it means they're being carried on a plasmid. If they're blue, it means they're integrated in the genome, okay? And at the same time, of course, we can see these phages too. So here's an experiment, right? This is D stands for donor. B is baseline, that's your patient who has been sitting on antibiotics for however long, awaiting the fecal transplant. And then 1 week, 2 weeks, 4 weeks after the FMT, after the transplant. And so what you can, and of course now we can watch it not just sort of the traditional metagenomic way, but we can see the mobile elements, right? So you can see the donor had a very rich microbiome, very little AMR, right? Whereas the patient, you can see their microbiome is basically wiped out, and then it gets recovered by the transplant. But of course, what's cool is one of the interesting, we don't really know what to do with this yet, is we can see this observation, even though this patient got better, it still has this one organism that's just carrying a ton of AMR. So this is an AMR reservoir that's going on inside of this patient. And simultaneously, we can say, okay, well, here's here's what the phages are doing. We can tell sort of which phages are infecting whom. Of course, the goal here is to try to find the, maybe causative phages, maybe phages that can be used as biomarkers or therapeutically to impact some of these clinical outcomes. And of course, this is not limited to human, right? We're doing these things on a large scale. We've done hundreds, possibly thousands of these samples by now. If you go into, if you work on human, most of your MAGs and most of your things of, the MAGs are relatively well studied because, human poop samples have been studied very heavily. But as soon as you go into like animals, the environment, water, et cetera, everything's novel. So it's a really good way of getting to those novel genomes that you just can't get any other way. And so, we sell this stuff. So we have kits, we have services, we have a platform. The software is all automated, everything I've shown you. And so just in the last minute or so, I'm just going to tell you about a project we're doing. So as I've shown you, right, like this one thing that this technology brings is the ability to extract phage host interaction data from unculturables, from wild things in nature, without kind of extreme efforts. So here's this like pig sample. You can see there's so many phages in here that you can barely see the little dots, right? We can get hundreds of them, hundreds of phage host associations from a single sample. And similar to what the previous speaker was mentioning, the idea was, we're never going to be able to really culture everything in the environment or even scratch the surface because there's so many things out there. But we're thinking about approaching it from a big data way. If we have enough of these host phage interactions, we might be able to train a model. To recognize patterns, right? Can we use the power of big data like this to basically figure out the rules of what causes phages to choose hosts, and can we use that to, can we turn that to our advantage, right? You can imagine all sorts of ideas, many of which were mentioned in the previous talk, right? Everything from, predicting, having predictive models of mobile element host range, everything to developing new phages to fight off resistant organisms, to look for new antibiotic resistance AMRs, antibiotic resistant reservoirs, find a way to defense against biothreats, look for new CRISPRs, all that kind of stuff. And so we've got funded by Gates Foundation and NIAID to basically generate the world's largest phage host interact on database. Technically, I think we already have it, but we're building it more. And so we're looking for people who are be willing or interested to contribute samples. We decided to do this in sort of a grassroots way where we, instead of kind of hoarding our own samples, we would kind of talk to folks in the community and say, if you have projects like this and you're interested in some free data, we can do this. So we're looking for samples kind of in the, colitis, IBD, FMT sort of category. We're doing a lot of wastewater and sewage samples from around the world. We're just doing healthy humans from different places in the world, different environments. The goal really is to generate as complete a data set or as diverse a data set of phages and their hosts as we can. So we're looking for all sorts of other understudied environments. This was a joke from a previous conference. Animals, all sorts of animal poops. We're doing corals, we're doing sponges, slime from caves, and I can just keep it going down. Like basically, we're interested a little bit in everything that could potentially add to more phage host interactions to our big AI model. And so that's it. And I don't know if there's any time to take questions, but I'm here.
All right, we'll have one quick question. And then, of course, if you have other questions, you can find Ivan in the poster session.
Hi, Ivan. Thank you for the presentation. I was just wondering, during the talk, you showed these kind of heat map figures where you have some promiscuous phages, but as well as co-infecting phages. Are those co-infecting phages or those sequences you're seeing actually like several phage particles co-infecting a single cell, or is it just like these different phages infect this bacterium in the whole community?
Right, that's a really good question. The question is, when we see on the heat map co-infections, are they actually co-infections, or is it just like one phage infects one cell and another phage infects a different cell? And the answer is both of those happen. And the reason why we can tell that is sometimes we can actually get a phage-phage interaction. So we can actually see that two phages are physically touching each other in some proportion of the cells. It's a very difficult, I mean, it's difficult question to answer dependably, but everything happens. So we, at some frequency, we see any permutation of things.